Okay, so I've taken these tubes out of the incubator. They've been shaking and growing overnight. You can see the pretty colors, so that's really exciting. Let's go through what we have here. So we have the positive control, that should be red. We have a negative control, that should be green. We have the LB, that shouldn't have any cells in it. That looks good. And then we have our 3 experimentals, and they don't all look the same. So we'll see what we get. So what I'm going to do is I have a sheet here all set up that matches my 96 well plate. And so I'm going to record which of these tubes goes into each of these spaces. And I'm going to pipette 200 microliters. So I'm just going to do the first one now, and I'll do the rest that you don't have to watch me. So I've got this set for 200. And I'm going to take my, this is one of the experimentals, so I'm going to take 200 microliters of the cells. And I'm going to put it into well A1, change tips, put it in the autoclave trash, and then I'm going to take another 200 microliters, put it in well B1, and then finally the third replicate, 200 microliters of the same tube in well C1. So you can see I've got those samples here. I'm going to write them down on my sheet, and then I'll go through and do the entire set. So here's one experimental group, here's the other 3 experimental groups. I'll do them all and come back in just a bit. Okay, so now I finished all the pipetting. I've got all the samples in there. So now we're going to go to the plate reader and read the fluorescence for GFP, RFP, and the absorbance at a wavelength of 600 nanometers. Let's go.